Alright, hi there AP Bio. Here is another lesson on DNA's central dogma. Today I am going to be talking about transcription, and if you missed the last one, it was DNA replication. It's not related to this, but this one you definitely want to watch before you will watch the translation one, which will be coming out tomorrow or the next day, we'll see. Hope you all are keeping busy somehow. I have a list of ideas of things you can do to stay entertained at the end here, so stay tuned for that. You can find this, hopefully, again, you got it before we left. It's all filled out and already uploaded on to Schoology. This is mine that I've filled out, and again, I've made just a few changes, so following along in the video can help you to make those adjustments. But other than that, I'm just going to kind of talk us through it. So, over here, for an overview, okay, if we are talking about going from DNA to RNA to proteins, that's what we refer to as the central dogma, and transcription is the process of going from DNA to RNA, or specifically mRNA. If this is a bacteria cell, remember no nucleus. There's also no Tata box, which we'll explain in a moment, and no transcription factors. It's just like, here's the promoter, start transcribing the gene, make the mRNA, make the protein. There's no barrier between these two processes since there's no nucleus, so it will immediately start getting translated by a ribosome into the protein. There's also no RNA processing, which you'll see is a new thing that we're going to talk about on the bottom part of this, probably new to all of you. So in eukaryotic cells, we have our nucleus. So DNA to RNA is transcription, but we're going to call that the pre-mRNA because it's not quite done. RNA processing is going to occur to kind of finalize what the RNA product will be. And then that final product leaves and again does translation with the ribosome out in the cytoplasm. So the nucleus is separating the processes. Here's another new thing. This pre-mRNA into RNA, part of the RNA processing is about introns and exons. Introns are going to get removed. Exons are going to get spliced and put back together. And then there's two more steps to RNA processing before it can leave the nucleus to go out for translation. So we'll talk about that at the end. So what I've got up here is a DNA molecule. It's not all twisted like a double helix, but it's just the parallel railroad track here. My 5' is up at the top and 3' on the right, and then the bottom side would be anti-parallel to that. So 5' on the bottom right, running this way to 3' on the left. If you're not recalling what that means, go look at the DNA replication video. So we know we have genes in the DNA, but where do we begin transcribing them? We don't want to start like right on top of the beginning and the end. So upstream of our gene, there's going to be a promoter, and just upstream of the promoter is what we call the Tata box. Kind of like Okazaki fragments, some of these words I just love. Tata box is one of them. The Tata box is just a code of DNA that's TATAAAA. That's just a specific sequence that RNA polymerase and transcription factors are looking for. So that whole region, this kind of teal and light green region, is the promoter, which is going to indicate where transcription should start. So RNA polymerase is our enzyme, this big play button triangle, that is going to be doing all of the work here. Unlike DNA replication that needed tons of enzymes to do all of the job, RNA polymerase is one like pretty smart enzyme. It not only binds, it unwinds, and it also brings in and assembles the incoming RNA nucleotides to be complementary to the DNA, so it does all of it. It may need help to get started, and that's what transcription factors are for. In the video I showed you about the mom's egg donating transcription factors and mitochondrial DNA, that's what I'm talking about here. These are proteins that assist in gene expression. They're helping RNA polymerase to bind to the right genes that need to be turned on at the right time. So they're kind of like, hey, come over here. Like, this is where the promoter is. This is where you need to be working right now. And this is the gene that we want expressed. So they're helping RNA polymerase to bind and initiate transcription. Downstream of the promoter, this pink region is our gene. So one gene could be hundreds or thousands of base pairs long, and the gene is going to be the information on how to make the protein. Notice I have put in some colors here. I'm going to talk about that at the end. Okay. The upper strand here is going to be our non-template strand, and the bottom one in the diagram is going to be the template strand. So the template strand is the one that the RNA polymerase will get built off of as a template. The other one is just going to move off to the side, out of the way as RNA polymerase works, and it's complementary to the template strand, and it's going to look really similar to the RNA getting built because it's complementary, but you have uracils instead of thymines, remember. So, but the non-template strand is just there to like seal up the DNA and keep that information protected and safe, but it will unzip and we're only going to copy one side. Which side it is determined by the promoter. So you will need to be reading 5' to 3'. Oh, excuse me. The thing about it, the RNA polymerase will read backwards. So it's reading the template strand 3' towards 5', but it's going to be building and assembling a new molecule in the 5' to 3' direction. So again, 5' being that part where the phosphate is sticking out, and 3' being the sugar part that's sticking out. So you're going to attach to the remaining sugar with a phosphate and keep going. The next part here, we're just elongating. Keep in mind, you've probably recalled this before, that uracil is getting used anytime thymine was going to be needed. So here's a TAC on the DNA, and we would have AUG on the RNA. So that's our pre-mRNA. Down here, we've got the rest. So I put RNA processing here. That's what this bubble is, so there's no reason to write it twice, but this whole bottom part is about RNA processing. So this might be the new part to many of you, unless you're in genetics, but this is what makes eukaryotes so cool. So let me zoom in just a little. Okay. So like I said, on the overview part, there are introns and exons. Introns, I know it sounds like you want to keep them in, and exons would be exiting, but that's wrong. Keep in mind, introns are intrusive or in the way, and exons are the parts that are going to get expressed. So I've banded my pre-mRNA, and I've labeled the exons as A, B, C, and D. And then this little cloud here is a spliceosome. So once we have coded the mRNA through the coding region of the gene, RNA polymerase will encounter this transcription terminator, and that's what's going to stop the whole process. This is not a stop codon. The start and stop codons that you might be used to are in the gene. They're part of the message that the ribosome is going to read, but the transcription is happening outside of those regions, upstream and downstream. So before I get into here, keep that in mind. That's a big mistake that students think transcription starts with start and stop codons, and that's not true. It starts with a promoter, and it ends with a transcription terminator. Once that is ended, RNA polymerase and the transcription factors detach, the DNA helix snaps back shut and twists itself, and it's back to normal. So the RNA processing is still happening in the nucleus. So that's what this region is here. We are basically having this complex set of enzymes called a spliceosome. You can see in your textbook, and off the top of my head, I don't know what chapter we're in the textbook, but you'd be able to find it. Central dogma. We're not yet on protein synthesis, but we're on kind of DNA into RNA transcription things. My textbook's in the classroom, so can't get it. So the spliceosome, it's going to basically like pull together pieces of the mRNA snip out the introns and let them fall away and reseal the exons back together. So here you can see I've resealed A, B, C, D without the introns. It could also seal them in different ways. A, B, and then D. B, C, D. A, C, D. I don't know if they can go out of order like A, D, B or anything like that, but this is one pretty fascinating thing. So let me mention that at the end here why. So spliceosome's doing its work. There's two more pieces to RNA processing, and that is the 5' guanine cap and the 3' poly A tail. Guanine being a couple of guanines up here at the beginning, and poly A being about 50 to 250 adenines at the end. The purposes of these I have on the side. It's preventing degradation. You've just kind of truncated and like snipped all this together, and you kind of want to put some filler on the ends just in case anything chews up or molecules get bumped off on the ends. So we're preventing degradation of the needed information. It's going to help the mRNA leave the nucleus, so that's going through a membrane here, probably about charge and polarity. And it's also going to help for the ribosome have somewhere to attach for translation, so it's kind of looking for this 5' cap. Only eukaryotes do RNA processing. Prokaryotes do not do this. So one of the really cool things about RNA processing and these alternative splicings of introns, I'm going to write that, or excuse me, exons. Alternative splicing, boop, and doing it in different ways, is that one gene can actually encode for multiple different protein products, which, if that's not blowing your mind, stand by. We noticed that there are probably about 25,000 something genes in the human genome. I hope I'm quoting that correctly. But there are not 25,000 proteins in humans. There's beyond that by a lot, like double almost. So we had this conundrum of like, how is it not like 1 for 1? One gene, one protein. That's what we thought it was. And we realized that one gene can actually manufacture multiple different proteins because of this alternative splicing in RNA processing by these spliceosomes. You can do different combinations of exons to create a different final product of the protein. So that's pretty cool. mRNA, this one, our messenger RNA, is now going to leave the nucleus for translation with the ribosome. That's the next video I'll be doing. On the back of this, there was just a couple of diagram pieces here. I think the one that is uploaded on Schoology has like an image of what the one nucleotide of RNA and one nucleotide of DNA looks like. This is just an overview, so keep in mind that any nucleotide has a sugar and a phosphate and a nitrogenous base, and to create this backbone and the uniform structure, we're connecting sugar to phosphate, sugar to phosphate, sugar to phosphate. The nitrogenous bases on RNA are just sticking out, but on DNA they're bonding across to each other. And we've got ribose sugar instead of deoxyribose sugar. That's where the RNA and DNA come from. Uracil is in RNA when it's needed to be complementary to adenine. Thymine in DNA, single-stranded double. And the last part here I like to think about is our entire process here. We're going from DNA to RNA as transcription. We're just rewriting the message because the language is the same. We're still talking in nucleotides, so it's like writing hello in print and then hello in cursive. You just sort of like modified it a little, but it's still the same language and same thing there. Where translation, we're going to change the language here and we're going to be turning it into amino acids, and that's where we're now in a new language of like hola or bonjour or dzień dobry. And so that's where we'll be going next. I have something in mind where we can upload messages in DNA to each other on Schoology and then decode them. So I'll work on something like that, like a little puzzle for you guys to solve in the meantime and practice this. And as soon as I finish translation, I will go ahead and upload some formative assessment on the AP classroom, so you've got something to work on and work your brain. But in the meantime, I thought about some ideas to keep you busy. So on my Instagram, I don't remember which company it was, but they're giving away paint by number canvases for free right now, so check that out. You could maybe learn to bake bread. It's complicated. You've got a lot of time on your hands right now. Why not become a baker? Origami. Meditative drawing. If you enjoy just kind of like quietly drawing and doodling, you don't have something to do, look up Zentangle or mandalas. Those can be really kind of repetitive and meditative ways to kind of help reduce some of the stress or anxiety you might be feeling right now. I thought journaling would be pretty cool to pass on a daily log of what you're doing to your grandkids during this unprecedented time. Build a model or Legos, play with little brothers and sisters and stuff and build big fortresses. I don't know. Overdrive is the online library app. I know the libraries are closed, but you can still get the Overdrive app and log on to King County, Seattle Public Schools Library, and you can get audiobooks and digital downloads there and kind of keep expanding your brain. And couch fort, why not? I feel like building a fort and watching a movie tonight with my husband. That sounds like fun. So hope you're staying entertained and not losing your mind too much yet. All right, I'll see you next time with translation. Bye-bye.